Hi everyone. As we begin to prepare for the AP Bio exam, I thought that I would give an abbreviated version of each of the units, just kind of hitting the highlights. And then if you're struggling with any one particular topic within this, then maybe go back and watch the more detailed version of the PowerPoints that I've recorded before. So Unit 1 is all about the chemistry of life. This was the first thing we learned, so it's probably the longest time ago, and chemistry is not everyone's favorite. So things that we learned in this unit include water and all of the properties of water and the importance of hydrogen bonding. We learned about the other elements of life, and then we dove in on the 4 major types of macromolecules. So, elements of life, always remember CHNOPS: carbon, hydrogen, nitrogen, oxygen, phosphorus, and sulfur. Those are kind of the most important elements for us in life. The first 4 are making up, 96% of life, and then we have other trace elements. So the thing that makes something organic is carbon, meaning life. If it doesn't have carbon, then it cannot be alive. Quick refresher on atoms. This might come into play, it might not, but just know that an atom is the smallest unit of matter that actually maintains properties of an element, and the elements are the things that you find on the periodic table. Atoms are composed of 3 major things. There are protons and neutrons that live in the nucleus or the center of the atom, and then there are electrons that float around the outside. And really, in terms of biology, we care about those outside electrons. They're called valence electrons, and those are what can form interactions in order to create bonds between different molecules. In terms of bonding, one that we talked about a lot is covalent bonding. Covalent, co means together. So covalent bonds are sharing valence or outermost electrons. And they can be in 2 forms. They can either be polar, in which case they are not shared equally. And then there is non-polar, which means they are shared equally. So non-polar is pretty easy. In order to share things equally, those are usually bonds between elements that are the same. So O2 or H2. A lot of gases don't exist just by themselves in the atmosphere. They like to partner up. Whereas polar, think of like, siblings close in age. They don't necessarily like to share equally. So there is usually one that is more electronegative, just meaning it has a stronger pull. It's the stronger sibling of the 2, a stronger pull on electrons. And so then because of that, you get kind of a little bit of a difference in charge. So here's the 2 picture examples. Again, we talk about water a lot because that's, a super important thing for life. So you can see that the oxygen is pulling the electrons closer to it. So it ends up with a more negative charge because electrons are negative. And then we end up with the H's that are a little bit more positive, whereas it's equally shared among similar molecules. Ionic is different. We don't talk about this too much in biology because biology is mostly covalent and hydrogen bonds, but ionic is not sharing. So one just straight up steals it from the other. The one that is going to take is the one that becomes slightly negative, and the one that gives it up is the one that's slightly positive. Remembering electrons are negatively charged. So when you give one up, you become more positive. When you gain one, you become more negative. And these really occur when you're combining metals and nonmetals. The goal of an atom is to have 8 magical valence electrons. And sometimes you need to gain 1 or 2 to make that happen. And sometimes it's easier to lose 1 or 2 than to try to gain, 6 or 7. So the goal is to get to that happy number, and metals and nonmetals come together oftentimes to make this bond. And it is really, really strong. In terms of biology, we love our hydrogen bonds because that holds a lot of things together, whether it be molecules of water, There are hydrogen bonds located between the base pairs in DNA. There are hydrogen bonds located in the structure that's holding amino acids together when proteins fold up. So we see them a lot. And really what happens is in a water molecule, the slightly negative charges on one side of water molecules attract to the positive charges of the hydrogen molecule on another, or on another water. And so then those form a hydrogen bond. So within the water molecule itself, there's polar covalent bonds, and between water molecules, there's hydrogen bonds. It doesn't always have to be water. So like on the right, you can see it's an example of ammonia. So anything that is polar can form these types of bonds between the positive and negative parts of those molecules. There is a review chart just kind of giving you a quick review of those three things. So on the left, we have covalent, on the middle, we have ionic, and on the right, we have hydrogen. And just the collective importance of all of these things are made or broken by chemical reactions. Okay, so water and all of its amazing properties. We already know that it unequally shares. We already know that it makes hydrogen bonds. Because of the way the molecule is set up, it can actually form 4 different bonds, and you can kind of see it makes sense. There's one off each hydrogen, and then because of the extra molecule or electrons on oxygen, it can actually have 2. Okay, so surface properties for water. Cohesion, again, co means together. So water molecules are going to stick to each other. That is what gives the attractive force of surface tension, where it's kind of a function of how hard or how much effort needs to go in order to actually break a bond. And then that brings in things like bugs that can walk across water because they don't weigh enough to break that surface tension. Other important property of water is adhesion. So adhesion is bonding of unalike molecules. So think of adhesive tape. You're sticking 2 things together. And here we have water. But one of the major pieces of adhesion and why it's so important is the idea that you can have water move up a tree, for example. And that is what allows water to move vertically upward, kind of against gravity. That's the force of adhesion, and leads to the idea of transpiration. So the water molecules are sticking to themselves, but they're also going to be sticking to the parts of the plant called the xylem, and then it's moving vertically upward, and then eventually it can make it to the upper leaves and branches, and then out and dissolve into the air. Another property of water is that it has a high specific heat, so it doesn't change temperature very easily, which is why in a warm day it's still possible for a large lake to be cold because it has not absorbed enough heat. It's another reason why lakes don't completely freeze over, because it can help contain a lot of that warm temperature underneath. And then us too, like we are 65% water. So it helps us maintain a stable temperature because we don't fluctuate so easily. Evaporative cooling. So we know that we sweat in order to make ourselves cooler because the act of evaporation sucks heat off of our body to make that happen. And then plants do the same thing with transpiration oftentimes. We know that ice expands, or water expands when it turns into ice, and that can form layers in aquatic environments. So therefore, the entire lake doesn't freeze. Water is the universal solvent, meaning that it dissolves a ton of stuff. So remembering vocab, the solute is the thing that dissolves. The solvent is the thing that's doing the dissolving, and then it creates a solution. And so the idea is water is super versatile. So it can dissolve a lot of things, pretty much all polar substances. Okay, We learned about hydrophilic and hydrophobic. Hydrophilic is water-loving. Hydrophobic is water-fearing. So on the left, that means they love water. Often they're going to be polar. Things that are hydrophilic include salt, sugar, cellulose. Your blood is actually hydrophilic. Hydrophobic, not water, doesn't like to mix. So that's where you can think of things like oils and fats. We have a phospholipid bilayer, if we remember, around our cells, the cell membrane, and it is composed of both hydrophilic and phobic regions that we'll kind of get through later. Just understanding structure function. So depending on how the elements are laid out in a particular molecule, they might carry out a specific function, and oftentimes if they have a similar shape, then it might affect its function in similar ways or slightly differently depending on what's going on. But here are two examples of molecules that have similar structure and therefore can trigger similar function by activating receptors in this case. Another property that has to do roughly with water has to do with acids and bases. So acids are ones that have hydronium ions, meaning they gain protons. So a water molecule takes on an extra proton and it becomes acidic. Whereas if a water molecule loses a proton, it becomes basic or a hydroxide ion. I would kind of think of it like the number of H's is really the measure of acidity. How many protons something has. If it has a lot, then it's an acid. If it doesn't have a lot, then it is a base. And in terms of the pH scale, anything under 7 is acidic, anything over 7 is basic, and then 7 itself is considered neutral. Here's just an example of different things that fall in that different category. So for us, like our intestines are around 8, but our stomach is at 2. So depending on where you're looking at in your body, you can kind of get an idea. We do have the idea of buffers, which are substances that try to minimize change in pH, because it's not really a good thing if we change pH all the time with large swings in fluctuation. So what that usually means is that there are weak acids or bases that can kind of absorb or release some of those protons, and it prevents large swings. So, for example, we have buffers in our blood that try to keep our blood pH around 7.4, because if we swing too high or too low, then we die. Those kinds of things. Real world application, pH is important. If, for example, the ocean gets too acidic, there are greenhouse gases in the air from emissions, CO2, that carbon dioxide can combine with water and eventually produce carbonic acid, which is bad because as acid builds up in water, coral reefs begin to die, and then we kind of think of the ecological consequences of that. So that's just kind of one application of where pH matters too. You do have a slide somewhere that kind of covers the different properties of water and examples. So you might be asked a question related to water properties, thinking about cohesion, adhesion, surface tension, specific heat, evaporation, and then universal substance or solvent. Okay, so the other half of Unit 1 was really dedicated to all of the biomolecules. You need to know how they come together, the 4 major categories, and kind of their general properties. So, quick refresher, there are monomers and polymers. Mono means one, poly means many. So a monomer is just going to be the individual unit of these macromolecules, and then a polymer is going to be when we start to link them together. And oftentimes, depending on how you stick them together, you can end up with a lot of resulting shapes. So again, we're tying it back to structure and function here. the way that you put the monomers together determines how the polymer comes together and therefore can then affect its function. I'm guessing the AP test is going to be a lot of big picture topics. So the idea of structure function is one of those. How do we put things together? Things come together via dehydration synthesis. I always think of it like dehydration synthesis is two words. You're putting two things together. Synthesis means to make. So we are making something by dehydrating it. And when you're dehydrated, we're taking away water. So as you can see in the picture, in order to link things together, we remove a water. There's an OH on one of the molecules and an H on the other. And when you take them away, you've cut out a water molecule. And then that can open up a bond formed between two monomers. All the different macromolecules that we're talking about get linked together in this fashion. So whenever we're putting things together, it's always dehydration synthesis. And on the flip side, if we take away water, we can link things together. We can break things up by adding water. And this is one word. So it's one word, one thing that will get broken into two. Lysis means to break. So in this case, hydrolysis literally translates to breaking with water. And the OH just gets plugged on back onto the one end and the H onto the other. All right, nucleic acids. So key parts of nucleic acids include a phosphate, a sugar, and a base of some kind. So you can see the nucleotide here is kind of the base unit of a nucleic acid. And so there's the phosphate group, and the backbone of DNA or RNA is alternating phosphates and sugars. That sugar depends on if you're talking about DNA or RNA. And then there's a nitrogenous base. So those are your A, T, C, or G. And this slide just kind of highlights that. A, T, C, and G, there are purines, which are A and G, which are bigger. And then there's pyrimidines, which are C and T, and those ones are a little bit smaller. But those are nitrogenous bases, and you can tell by the presence of nitrogen and then the phosphorus. So key elemental parts of a nucleic acid are carbon, hydrogen, but then you'll also see nitrogens, oxygens, and phosphates. Other macromolecules don't always have that. The sugar could either be deoxyribose or ribose. Essentially, the only difference is this one oxygen, whether it's present or absent. And then in terms of joining together, the phosphate would join here at the 5' end, and then there would be another one down here joining to this molecule, and then the nitrogenous base would be over here. Those come together via a phosphodiester linkage. So I always think of like phospho because it's got phosphate, di for two, and in this case, it's an ester bond because of this oxygen, but I don't think you really need to know that. Nucleic acids are put together on the backbone by phosphodiester bonds. And then the bases, as we will learn, are put together by hydrogen bonds. When we want to link them together, we do a dehydration synthesis reaction. We remove a water, and you can kind of see how that happens. Key features of DNA and RNA, like I said, the sugar, there is a difference in terms of a base. So DNA has thymine, RNA has uracil, otherwise the other three are the same. And then DNA makes a double helix, while RNA makes a single helix. I guess you can't call it a helix, it's a strand. All right, the next type of macromolecule is proteins. Pretty much all the functions that are done in a cell are done by proteins. They come from a large chain. So the polymer is called a polypeptide. They are formed from the linkage of individual monomers called amino acids. So we have 20 base amino acids that you can link together. And you can see here they all have the same basic structure. And then they have a different R group attached that can give them different properties. So the order in which we link these amino acids can then determine overall structure because they have slightly different properties, and that means that they're going to fold a little bit differently. And then depending on how they fold will then determine their function. So here's an example of the final structure of a hemoglobin. For example, it's got 4 kind of polypeptides twisted and clumped together. And in this case, it's also carrying the iron component. So you can kind of see what the 3D structure looks like. Every amino acid has the same basic parts. It's got an amino group. So when you're thinking about the elements that compose protein, it does have carbon, hydrogen, oxygen, and nitrogen. So there's an amino group, and there's a carboxyl group, and then there's always a hydrogen. The part of variability in an amino acid is the R group. The graphic got a little messed up in this case, but depending on those R groups or their side chains, they can have different properties. Some of them might have nitrogen, some of them might have charges, some of them might not have charges, some of them might have structural rings, and so on. They are linked together by peptide bonds. So again, this is still going to be dehydration synthesis, but the carboxyl group, so there would have been an OH over here, and the amino group of the next amino acid has an H over here. Like so, you can kind of see, gets removed, and then we form a peptide bond. So amino acids are linked together by a peptide bond to form a polypeptide that when you fold together becomes a protein. I hope that made sense. All right, so just the chain itself is called the primary structure. So just linking with peptide bonds, one singular super long chain is called a primary structure. The head or front, so to speak, is always the amino group, and the tail is always the last carboxyl group. The secondary structure is when those those chains fold into either an alpha helix or a beta pleated sheet. So this is where hydrogen bonds come back in. Depending on whether they or where they are joined together, you will result in one of those two structures. So that's called secondary structure for kind of the second level. From there, we start to get R groups doing their thing. So R groups can be hydrophilic, they can be hydrophobic. There's all kinds of things. They can form special disulfide bridges, which is what the cysteines do because they have sulfur, and cysteines really want to stay together. So when you're going from an alpha helix or a pleated sheet, the R groups start to interact and kind of fold, and then you end up with a 3D-like structure called tertiary structure. And then on rare occasion, you can have more than one of them coming together. Similar to hemoglobin that had 4 chunks, that would be called quaternary structure. Quart means 4 in this case. And when I said that, it's supposed to, quaternary. But not all proteins have to have that high level of structure, just so you know. Proteins can be tons of things. They can be enzymes. They can be antibodies. They can be storage proteins. They can be transport proteins. They can be hormones. They can be receptors. They can be contractile proteins like actin and myosin. They can have structural support like collagen. So the idea is that proteins have a ton of functions. If it's performing a job in your body, it's probably a protein. We can destroy proteins, however. That is called denaturation. If a protein is denatured, it has been rendered inactivated or irreparably damaged in some way. And in order to do that, you can go outside of normal pH ranges. You can go outside of normal temperature ranges. You can mess with the salt or possibly other environmental things. So every protein kind of has a sweet spot for all that. And when you go outside those ranges, that's when you start to get problems, which is why we can't have fevers super, super high, because then our proteins start to denature. Or if our pH range gets way out of whack, then things in our blood, for example, can't perform their duties. All right, lipids. Lipids are fats. They don't really form polymers. They form fatty acids or triglycerides that we'll talk about. They form the phospholipid bilayer. And then the weird looking section is also, in this case, steroids. Key thing about lipids is that they're not polar. They hate water. So therefore they are hydrophobic. They are made mostly of hydrogen and carbon. So you won't be finding nitrogen, you won't be finding phosphates anywhere. It's carbon, hydrogen, and then at the very top, you're going to get some oxygens. Because that's where what will be the glycerol molecule forms, and then these long chains are all different hydrocarbons, meaning carbons and hydrogens only. So at the top here, you can see the glycerol molecule, and that's what is used to form a triglyceride, and it's called that because it has 3 molecules here. So in that regard, there are a few oxygens, and then you can see the long chain hydrocarbons. That's why in this regard, it's not really forming a polymer from a monomer. It's kind of the linking of these things together. And it's called an ester linkage, again, because of this double bonded oxygen. It's called an ester, similar to the phosphodiester bonds to link the nucleic acids together. They still link together by dehydration synthesis. So in that regard, you can consider that a similar feature. They can be saturated or unsaturated, and therefore it results in them being solids or liquids, and it can then determine their function as well. So the saturated ones, saturated just means that it is saturated with hydrogens. Every single one of the carbons in those long chains has hydrogens attached to it via singular bonds. And because of that, these chains are all perfectly straight. So they can pack together really closely. And because they can pack together really closely, then they are going to be solids at room temperature. So butter is one example. Unsaturated, on the other hand, is missing some hydrogens. So because of that, to make the carbons happy, instead of a single bond, they might form a double bond or a triple bond, even depending on how many hydrogens are missing. And those are unsaturated. It also forms a kink. So when you're adding in extra bonds, they're not straight anymore. And that kink or bent structure means that these chains can't pack quite as closely together as they had before. And that's really what makes them liquids at room temperature. So olive oil would be one great example. Phospholipids, which are composing our cell membranes, have 2 regions. They do have a polar part, which is the phosphate part. They have a phosphate group up here. So here's the glycerol. You can see the phosphate attached to it. And then, because of that, it's a little bit polar. So this circular part loves water. But then we have these tails stuck onto the end. Those are still nonpolar, still not liking water. In this case, it's unsaturated, and it will be the basis of forming phospholipid bilayers for cell membranes. All right, carbs. Everybody loves their carbs. So sugar. Carbohydrates are sugars. The singular unit of a carbohydrate is called a monosaccharide, or a simple sugar, a one sugar. When you start linking them together, they are called polysaccharides. So the main component, or the main heavy hitter monosaccharide, is glucose. And then you can start to link it together to make disaccharides and so on, like sucrose, that's table sugar, and other things. The formula for glucose is C6H12O6. So by definition, all carbohydrates are in some multiple of CH2O. Those are the three elements that compose a carbohydrate. You will not see nitrogens. You will not see phosphates. Traditionally, they have this kind of structure here. They don't always have to, and it might be represented in vertical form, but this is the ring structure of a glucose molecule. But we also know that there are sugars like ribose that only has five carbons. If it ends in ose, that's a good giveaway that it's a sugar or a carb. They are joined together by glycosidic linkages. So it's still dehydration synthesis, but that's what they are formed and how they are formed. We learned this later, but when you're thinking about how they are linked together, there is directionality in a lot of the things that we do. So how things join together, here's an example of that carbohydrate sugar. In this case, it's deoxyribose. You can see the alternating phosphates. We have talked about 5' and 3' ends in a later unit. But because of that, it oftentimes creates directionality when we're forming these things and can lead to specific rules later when we're doing things like DNA synthesis. So just keep that in mind. This I already covered. I'm not sure why it's here. Monosaccharides, similar to other things, depending on how you put those together, can form different things in terms of structure. And that's why we can have glycogen, which is more of like a long-term storage, and so on. And you know this already, hopefully. Change in structure then causes a change in function. Big picture topic to think about. At the very, very end, review. So amino acids, what they are used for. So enzymes and all different kinds of proteins, what their functions are. Nucleic acids, the base component is that nucleotide, DNA and RNA, and then their functions. So these are good slides to kind of pause for a moment and make sure you have all this material absorbed. Carbohydrates and what they're used for. And then in the last category, we have lipids and what they're used for. So take a moment and pause and see if you can absorb all that material. You've made it to the end of Unit 1. If any of this was super confusing or way too fast, please go back to the original narrated PowerPoints that go a little bit slower and a little bit more in depth.